So apart from just studying genes and proteins, you can use DNA technology to study DNA sequences themselves, which can mean studying genes, but it can also mean studying the sort of non-coding parts of the genome as well, and learning about those sequences. So there are a lot of techniques associated with this. One way to study the genome is using a genomic library. So, of course, a library, is a place where you have books that you can go read. So a genomic library is just a collection of pieces of DNA that you can sequence and study as individual units. So the way a genomic library is usually made is by starting with your chromosomal DNA. So you extract the DNA from your host cell, whatever you're interested in, and you cut the chromosomes up with restriction enzymes. So sometimes this is done with just one restriction enzyme. Sometimes it can be done with multiple restriction enzymes. But you'll then be cutting the DNA up into pieces of more or less random length and at more or less random locations. So any given piece may contain a gene or multiple genes, or it may contain no genes at all, or you may even have cut points within genes. But you take those pieces and they're all mixed together, and then you clone the pieces into separate vectors. So you use vectors that are compatible with your restriction enzyme, and you clone those vectors into recombinant plasmids, transform those into bacteria, and then each bacterial cell that picks up one of your recombinant plasmids then will be carrying a piece of your genomic DNA. And then you separate those cells out into separate colonies. The colonies, or the descendants from each colony, get into their own culture, and then you have basically a rack of little tubes, each of which contains a clonal population of bacteria that carry a piece of genomic DNA. Then you can extract the cloned cells, or the cells carrying your cloned DNA, extract the plasmids from those, sequence or do whatever you want with the genomic DNA. But then you have essentially your library is just that population of now modified bacteria. Now the thing about genomic DNA again is that it's just all the DNA. So that includes the genes, but also the DNA sequences between genes and other non-coding bits. So if you're only interested in the coding part of a genome, the parts that actually code for proteins, instead you would make a cDNA library. And this is, again, a collection of clones, but this time you're making the DNA, instead of just from the chromosomal DNA directly, you're making it from the mRNA. And the mRNA, you probably know, is the RNA copy of a gene that is used to make protein. So if you're making a library just from the mRNA sequence, you're only looking at the coding parts of the gene. So the mRNA, in this case, is extracted from a cell or a tissue, and importantly, not every gene is expressed in every cell all the time. So some genes are only expressed at certain times. Other cells are only expressed in certain tissues. So if you're making a cDNA library, it's kind of like a snapshot in time of just the genes that are being expressed in that cell at that moment. So you extract the mRNA from your source, and then you can't really make you can't use RNA for cloning. RNA itself just is not stable enough, and of course it can't be incorporated into a plasmid or replicated on its own. So the mRNA has to be converted back into DNA. So the way to do that is first combine it with a oligo-DT primer. So the reason to use oligo-DT is that most genes, when they are transcribed at the end of the RNA sequence, and we'll talk about this later on when we talk about transcription in more detail, but always at the end you have this chain of A's that gets added after transcription. So this isn't part of the gene. That poly A tail is not part of the gene. It's just what gets added to the end of the RNA after transcription ends. So because most genes have this poly A tail added to the mRNA, you can use a poly T DNA primer that will bind to that poly A tail. that acts as a binding site for an enzyme called reverse transcriptase. And reverse transcriptase does pretty much exactly what it sounds like. So transcription is the process of converting or making an RNA copy of a DNA strand. Reverse transcription would be making a DNA copy of an RNA strand. So there are enzymes that do this. They're actually derived from viral enzymes. So there are viruses that actually carry around their RNA. Interestingly, the coronavirus is one of these. So the coronavirus actually carries around an RNA copy of its genome, but also instructions for making a type of reverse transcriptase enzyme, which will make a DNA copy of its genome so that its host cell, one of your cells if you're infected, can then make more copies of the virus. But in this case, we're using reverse transcriptase to make this mRNA-DNA hybrid. Once we make the DNA copy of the RNA, we don't really need the RNA anymore, so that gets degraded. And then we use a primer with another oligonucleotide to make a primer at the 3' end, or bind to the 3' end of our DNA. Now here we have to know a little bit about what's at the 3' end. And in that case, but then if we don't, we can always, there are ways where you can essentially attach a 3' sequence to the 3' end of the DNA using a DNA ligase. And then you do PCR, essentially, or use a DNA polymerase to now make a copy of that original strand. And so then you're left with this duplex DNA, one strand of which will will be complementary to the mRNA. The other strand of which, because it's complementary, will have the exact same nucleotide sequence as the mRNA, which means it will have the same nucleotide sequence of the gene, but just the coding part of the gene. So it doesn't include things like introns. And then you can take that structure, which here we call this what you just made, the cDNA. C actually means complementary, but you can also think of it as the coding DNA. So reverse transcriptase is used to make the cDNA. You put the cDNA again into a cloning vector, and then you transform the cloning vector plasmid into a bacterial host. The host grows, and so every clone of that host cell, bacterial cell, will then contain one of the mRNA sequences. So then you have that collection of bacteria becomes your cDNA library. And again, each one can be, each library represents a different situation. So again, you might look at, you might go back and see which genes were being expressed in a cell, perhaps at different times. So if you're studying development, for example, you can look and see which genes are being expressed in the cell in an animal as when it's an embryo versus when it's an adult. You can compare gene expression in a tissue before and after, say, a drug has been introduced, or you can make whatever comparison you want. So that's the power of a cDNA library. Now, the process of genomics, meanwhile, which relates to having a genomic library, is just the study of whole genomic sequences. So you're not necessarily just interested in individual genes, but you're interested in the whole genome. So that includes the genes, but also all the non-coding parts of the DNA. All the DNA of every chromosome in that individual, which again should be the same for every cell in your body, and should be the same or pretty similar to all the members of your species. And so by looking at the whole genome, you can look for information that you wouldn't otherwise be able to get just by looking at individual genes. Now, genomics is a pretty new field because genome sequencing is a pretty new field. You can kind of see over here how genome sequencing has progressed. So the first genome ever sequenced was of a bacteriophage. So this Phi-X174 is just a type of bacteriophage virus. Its genome was sequenced in 1977. And so then the first actual genome sequencing, and so that can be done with a virus because they have relatively short genomes. But the first attempt at sequencing anything larger than that came much later, so in 1990. And in fact, the first non-phage organism ever to be sequenced, its genome, was a bacterium, H. influenzae, and its genome was sequenced in 1995. Now, after that, the sequencing of genomes became much more common. You have sequencing of yeast, the sequencing of archaea. E. coli's genome was finally sequenced in 1997. E. coli is a common organism, again, as a common bacterial strain used in research, so that was useful. Again, C. elegans, that's a nematode worm. It's commonly used as a model animal in a lot of different research fields. Not long after that, you have fruit flies. So nematodes and fruit flies both are important host organisms or model organisms for research. C. elegans was the first animal, in fact, whose genome was sequenced, and the fruit fly shortly after that. Finally, in 2001, the human genome, or a draft of the human genome, was produced. The Human Genome Project was this enormous project. It actually started about 10 years before this. It involved several different labs, several different groups working at the same time. Again, we talked about DNA sequencing in the last chapter. We talked about Sanger sequencing. Of course, sequencing whole genomes requires a lot more sophisticated techniques than what would have been available in the early days of DNA sequencing. But starting around 2000, it became easier and easier and faster to sequence entire genomes. So again, human genome, first draft in 2001. The complete human genome didn't really come out until 2003. And so it took a couple years to kind of finalize. But again, that whole project took about 10 years or so. And in the 18 or 17 years since then, you've had a lot more species sequenced. Over 2,000 bacterial genomes have been sequenced starting in 2010. The relatives to humans, the chimpanzees and bonobos, were sequenced. Also, extinct species can be sequenced now. So DNA has been extracted from, for example, Neanderthal. Neanderthals are the closest relatives to humans, but they went extinct about 8,000 years ago or so. But their remains from Neanderthal have been found that are intact enough that you can extract DNA. So we have now in 2012 the complete Neanderthal genome. Other hominids like the Denisovan hominids have also been sequenced, and relatively recently, the Siberian woolly mammoth, which also went extinct several thousand years ago, its genome has been sequenced as well. And now, where again the first human genome took many years and many millions and probably billions of dollars to sequence, now the speed and the cost of genome sequencing has gone way down. So now it's technically and financially feasible for individuals to have their entire genome sequenced for, in some cases, less than $1,000. So we're not actually that far from the idea of personal genome sequencing. So services where you could just send your DNA sample and have your entire genome sequences. There are already, in fact, services like you've probably heard of, like 23andMe. Now those services don't automatically or don't necessarily sequence your entire genome. They just look for certain genetic markers in your DNA, which doesn't require sequencing the whole thing. It may just require sequencing small segments. But again, we're not that far away from the day when it will be relatively routine for people to have their entire genome sequenced. Now, what can you learn from genome sequencing? It depends on the genome, but first of all, the genome itself doesn't actually tell you much. So if you just look at the genome of your own genome or genome of any species, all you're going to see is a long stretch of bases, just A's and C's and G's and T's, which is pretty much useless unless you know what you're looking at and know to look for certain sequences that define, for example, genes that code for protein. And so the process of genome annotation is the process of looking through a genome and identifying important genes or regulatory sequences. Now, no one actually can do this manually. You can't look at a chromosome that's many hundreds of millions of base pairs long, and actually just see patterns that tell you information about a gene. So instead, genome annotation is largely done by computer. So once a genome is sequenced, the genome sequence is stored on a computer. A lot of genome sequences are publicly available actually on web servers, and so you can go and download most genome sequences. And then you can either write a program or use one of many available web-based programs that will look through genome sequences for you to try to find. You can either look for individual genes, or you can upload genome sequences that you have, or gene sequences that you've identified or sequenced yourself, and have it search through a known genomes to try to identify what you are looking for. And so the Human Genome Project in particular has revealed quite a few interesting facts that were surprising at the time. For one thing, only 1.5%, really just almost a tiny fraction of the human genome actually contains protein coding genes. So these are actual genes that code for proteins and are functionally expressed in the human genome. So the entire human genome, less than 2% of it actually codes for proteins. The rest of it is all non-coding DNA. Now, we used to hear the term junk DNA used to describe any non-coding DNA. We don't really use the term junk DNA anymore, because it turns out a lot of these DNA sequences, even though they don't code for DNA, do have recognizable patterns and seem to have, at least in some cases, some important functions. So for one thing, and you probably know this, in eukaryotes, a gene consists of two types of DNA segments. The exons are the part that actually get translated into protein. And so one gene usually will consist of 4, 5, 6, 7, a number of different exons, and then several introns. And so the introns are segments of the gene that get transcribed into RNA, but then before the RNA gets translated into protein, those introns get spliced out. So there's a whole complex of enzymes that look for those introns, and they essentially chop them out. And then so then the exons are the only parts that are remained, and those are the only parts that actually get translated into protein. So it turns out, in fact, that there are way more introns than there are exons, or at least the amount of DNA dedicated to introns, or that consists of introns, is a lot larger than those involved in exons. And then, but even the introns and exons together still only make up around a quarter of the entire genome. The rest of the genome consists of these regulatory chromosomal regions like the centromere and the telomeres. Those are important for cell replication, because when a cell replicates, the centromere and the telomere are structures that are important for moving the chromosomes around within the cell during the processes of mitosis and meiosis. Some of these are what are called segment duplication. So sometimes when chromosomes get copied by mistake, part of the chromosome will get duplicated, and sometimes those duplications lead or are bad, and so they cause the cell to die. Other times they just stick around. So sometimes these duplications end up as permanent parts of the genome. And then there are simple sequence repeats, which may or may not have a function. And then there's a whole bunch of the genome, almost half, that consist of these things called transposons. So transposons are interesting because, again, they don't seem to code for any DNA, but there are enzymes that are encoded by certain parts of the genome that will actually cut these transposons at certain points in time and move them to other parts of the genome. So that's why they're called transposons, because if you look at even closer related individuals, you find that these transposons can be in different places. So these transposons move around within the cell, can even sort of copy themselves within the cell. That means that, or within the genome, that means that one transposon may show up more than once in the same genome. And so these seem to make up a big chunk of the genome. Again, it's not clear if they actually have any function within the genome, or if they're just sort of simple genetic parasites. They've just figured out that these sequences, because they get copied and moved around, they just sort of live, quote, within the genome. But in any case, the fact that, again, only 2% of the whole genome actually involves protein coding sequences is pretty surprising. Again, some of these other sequences have functions that may include the expression of those protein coding genes. So even in an mRNA, after you remove the exons, you still have these regions called untranslated regions. So even for a regular gene, there's a 5' untranslated region and a 3' untranslated region. Those still get spliced together with the exons to form the mRNA. But they're called untranslated because the ribosome doesn't recognize them and doesn't translate them. So translation for a gene always starts at the first exon, at the boundary between first exon and the untranslated region, and then translation stops at the last exon and the boundary with the 3' untranslated region. So those regions are important. They're mostly important for translation itself. So the ribosome and the proteins that initiate translation recognize those untranslated regions. So they serve a function. They're just not themselves involved in or don't get coded into amino acids. And then there are a bunch of other regulatory sequences throughout the genome that are involved in transcription as well. And even within the protein coding genes, once you've identified the protein coding genes, you can look at all those genes and kind of classify them. Now, a lot of genes we know about because we know their function. So a lot of enzymes, for example, we've discovered through by studying the proteins themselves. But you can also find new genes or new proteins by looking at the genome. So if you have a DNA sequence for a gene, a known gene, then you can search for that genome sequence in the genome and look for other versions or sequences within the genome that are similar to it. So some genes have been discovered not by discovering their proteins, but by discovering their DNA sequences through genomics. And again, there's some interesting things to notice when you look at all the different protein coding genes. So enzymes seem to be one of the biggest categories of protein in the cell, which makes sense. Nucleic acid binding proteins are also important because those are the proteins that bind to DNA and control the expression of other proteins in the cell. But again, interestingly, there's a big chunk, more than a third of all the protein coding genes in the human genome have unknown functions. So these are not just DNA sequences that we don't know what they're for. These are proteins or protein coding genes that have no known function. Now, once you have the entire genome of an individual, of a species, then you can start looking at other aspects of that genome that actually have nothing to do with, may not have anything to do with genes themselves. One important type of marker that you can look for in a population of genomes, once you have multiple genomes from the same species, you can look for individual bases, locations within the genome where individual bases vary from one person to the next. These are called single nucleotide polymorphisms, or SNPs. So a SNP, or SNP, or whatever you want to call it, is just a position in the genome where the genome sequence is not the same from one individual to another. So of course, we're all the same species, which means that if you compare the DNA from two different people, the vast majority of the bases should be exactly the same. Every single letter should be in the same position and in the same order. But there are locations throughout the genome where there are just where you can have random mutations, and these mutations happen all the time. So every time a cell copies its DNA, the DNA replication system can make mistakes and put the wrong base in. Again, that's a mutation. Some of those mutations will be bad, especially if they occur in one of those protein coding genes. But again, protein coding genes only make up less than 2% of the genome. So most mutations will happen in those non-coding regions, and so that means they won't really do anything. So if a mutation happens in a part of the genome where you're not in a coding gene, or you're not in a regulatory gene, or even one of those elements like the centromere or the telomere, it won't really have any effect on the cell. So those mutations simply get passed down from generation to generation. In fact, you probably carry a few mutations in your genome that were not present in your parents' DNA. But you did inherit some polymorphisms. Polymorphism just means a difference from them that they inherited from their parents, and so on. And in fact, then you can look for SNPs in groups of people, or any members of the same species, and you can then ask questions about their relationships. So those SNPs can be markers, essentially, for groups of individuals that are closely related. In fact, a combination of SNPs that shows up in a given population we call a haplotype. So your haplotype consists of a combination of SNPs that are sort of define a group of people, again, based on some pattern of common ancestry. And usually when someone's studying populations, especially human populations, of course there are lots of SNPs in the genome, and although genome sequencing is cheaper and faster than it used to be, it's still pretty costly to sequence large numbers of genomes. So when people are studying genetics at this level, looking for these SNPs, rather than sequence the entire genome, there are systems that only look at a subset of SNPs within the genome, and those are called tag SNPs. So for any given haplotype, there's a small number of tag SNPs that are used to identify someone from being in that haplotype. So you can use these SNPs and the haplotypes to study relationships within a species. You can also look at evolutionary relationships between species. So you can compare DNA sequences across species, and again, even two different species should have common DNA. Humans and chimpanzees, for example, actually have over 90% of our DNA in common, at least in the coding parts of the DNA. So if you compare human and chimp DNA, you will find lots of bases that are the same. And those individual variations represent evolutionary changes, because the assumption is that at some point in history, but after humans and chimpanzees diverged, or humans and chimps diverged from our common ancestor, both lines picked up mutations that are now part of our modern genome. And again, you can compare lots of sequences this way and determine those evolutionary relationships. So, and so presumably the more similar two groups are, the more closely related they are. So chimps, gorillas, and orangutans, for example, in this particular gene, I'm not sure which gene it is, but their DNA for that particular gene is the same. Whereas if you look at other mammals, you see a lot more variability. And then you can then compare multiple sequences to figure out how groups are related. So for example, humans and chimps, we know are closely related to each other. They're more closely related to each other than either one is to any other living species. And we know that from looking at fossil evidence. We know that looking at anatomical homology. But we also can tell this by looking at their genome. So if we compare the genome of the human and the chimp, again, we can see that they are more similar to each other than either one is to any other species. But not only that, we can ask questions about what the common ancestor of human and chimps, what its genome might have looked like. So if you compare humans and chimps or any two species and look at the DNA for a given gene, you will find differences. And so if we compare this gene X, we compare the sequence of gene X in a human and the sequence of gene X in a chimp, and we find a difference. We find a SNP here. So this base here is a T in the human, and at that same position, it's a G in the chimp. The question is, which of those sequences, if it is either one, came from the common ancestor? So is that is that sequence the same as the ancestor? Or does the chimpanzee have the ancestral sequence, or does the human have the ancestral sequence? Or maybe neither one did. Maybe they both diverged from the ancestor. To determine that, you can compare the DNA sequence in that gene to another species that you know is still close to this group, but not not related to the ancestor between your two target groups that you're trying to study. So in this case, for humans and chimps, you might compare them to an orangutan. So if you look at the DNA sequence for gene X in an orangutan, if you saw that at that position there's a G, because it matches the chimp, or because the G is in the out group, the orangutan in this case is called the out group, then we could make the assumption that the ancestral sequence for that gene contain a G at that position. And so what that means is that the chimp lineage inherited that gene, and then it was a mutation in that gene that led in the lineage to humans that led to the modern human version of that gene. So again, these comparisons let you ask questions about the evolutionary history of different groups. And you can look, again, like I said, you can look at even for DNA of individuals that are extinct or species that are extinct. So there are, in fact, a lot of human species, species in our genus Homo, that lived not that long ago. So in fact, like I said, Neanderthals died out only about 8,000 years ago. And we have their DNA, and we've sequenced their DNA. And one thing we've discovered, in fact, is that there is DNA in the Neanderthal genome that actually shows up now in the DNA of modern humans. And in fact, what that implies is that at some point in after modern humans evolved, there must have been modern humans and Neanderthals that mated, and that's how Neanderthal DNA ended up in the human genome. But it's not in the entire human genome, which means that Neanderthals only mated with a subset of modern humans. In fact, you can only find Neanderthal DNA in humans that are of either European or Asian descent or Oceania descent, whereas modern humans of African descent generally don't have this Neanderthal DNA. So what that implies is that this mating happened after modern humans left Africa and went into Europe. And again, there's other kind of questions you can ask about lineage this way. Another important use for these SNPs, for these polymorphisms, is to study genetic diseases. So genetic diseases are ones in which a mutation in a gene or in the regulatory part of a gene causes the gene to malfunction somehow and lead to disease. Again, most mutations will do this. Most mutations in genes will prevent the gene from functioning, but what that usually means is the gene doesn't function at all, and if it's a crucial gene, then the individual carrying that mutation just doesn't survive. So in fact, most mutations are what we call embryonic lethal, which means that that you're never born with those mutations because you're not born at all. The embryo dies before it even develops. but every once in a while you'll have a mutation in a gene that doesn't completely is not completely lethal, and so that individual will be born with that malfunctioning gene, and that can then lead to disease. And so tracking down the gene that actually causes a genetic disease is sometimes difficult. So the first thing that you do to study a genetic disease is look at what's called a pedigree. So these diagrams up here are just drawings of the relationships of a family. Each symbol represents a person, and usually, I'm assuming in this case, the red diamonds are the people who have the disease, the white ones are the people that don't. Usually the line through a symbol means that person died. And so what this shows you is it lets you trace how that disease was passed from generation to generation. That will tell you some things like whether or not it's dominant or recessive. It can tell you whether it's on one of the sex chromosomes or not. But if you really want to find out where that gene is, you have to look for the gene itself. So you may be able to tell what gene is involved based on the disease, but if not, you can instead look for these SNPs, because the SNPs themselves don't necessarily cause disease, although sometimes they can. But if you can look for those SNPs and you can see which individuals have the haplotype that corresponds to the disease, you can look for the SNPs and use that to kind of zoom in on where along the chromosome the disease gene might be. So if all of the individuals, in this case that have the disease, for example, all inherit this combination of SNPs, this haplotype, then that tells you that the gene that's causing this disease may be in this part of the chromosome. So that is just a way to narrow down the possible culprits in a genetic disease like this. And finally, again, if you're looking at big populations of organisms, humans in this case, you can not just look at their evolutionary history, but actually their cultural history as well. So you can go back and see, when did especially humans have migrated and are living all over the planet. So you can look and see, when did those migrations occur? When did humans spread out over the whole world? And again, you can look at those SNPs, look at different haplotypes, and find where they exist in modern humans, and use that to kind of work backwards and figure out how they must have diverged. So in fact, we now know that modern humans first evolved, or at least the genetic ancestors of modern humans appeared somewhere in Africa, and then several migration events happened after that, and this is and then these populations sort of inhabited the rest of the world. And so you can see there have been various migration waves out of Africa over the about 200,000 years or so since modern humans first appeared. So for example, there was a big wave about 50 to 60,000 years ago into Asia, and that wave also went into North America, but then there was a second migration event into North America that was part of a more recent event about 20,000 years ago. So it suggests that at least twice humans have migrated from Asia to the Americas across what used to be the Bering land bridge. So now there's a strait of water in between Russia and Alaska, but there used to be a land bridge that connected the two, and so people just walked from one continent to the next. Now, of course, each of these migrations took hundreds or thousands of years to occur, but you can trace sort of the lineage of every human population this way. So again, that can kind of help answer questions about how people first arrived and how they may have brought, in some cases, cultural influences with them. All right, so that is basically the end of that chapter. So we are done talking about DNA for now. Again, we'll come back later and talk about DNA replication and things like that later on. But actually, next we will be talking about lipids.